Okay. Hello, everyone. Thanks for coming today. We'll get started in 1 or 2 minutes. Let me just get everything set up and we'll be good to go. Okay, so a lot of people are coming in now. Okay, that's good. All right, so I'll get started, and if anyone wants to join in later, they can also look at the recordings if they'd like. So anyways, hello everyone, hello Burst members, thank you and welcome to the second general meeting. We're really happy that you're here, and we understand that this week is all of your midterms or exams week, so we're gonna make this really worthwhile. We have really exciting announcements and a great general meeting speaker for you all, and a really nice post-meeting social, so stay tuned for that. But without further ado, we're going to do a little icebreaker. So this question is, what is a food that you dislike that most like or prefer? And you can drop down your answers in the chat box, and I'd love to read those. Ketchup, peanut butter, greens of any kind. Tyson, do you not eat vegetables then?
Nah, not at all.
Really? Someone said I'm so offended by these answers. Wow, this is a good list. Ranch, oysters, seafood, mushrooms. Oh man, I love mushrooms. They're really good with the food that I usually cook with. Tiramisu. Wow, we have a whole group of mushrooms going on. All right. Thank you for joining yogurt. Okay, so we're going to get started on presenting all the officers to you all. So if you haven't met any of us, we're really easygoing, we're really nice. We want to help you all out in your journey at Baylor. So shoot us an email if you want, or if you see us on campus, please do come say hi to us. We're really friendly and we want to get to know all of you. And so we're going to be showing a little clip from Kaplan. If you don't know, Kaplan makes all the test prep books, classes, and courses that are ranging from like ACTs to GREs, SATs, or MCATs. And so their representative has given us a 10% off for your next Kaplan order if you choose to buy one of their products. And you can just use Club 10 code to get that 10% off. And so let me get started with queuing up this video.
Hello, everyone.
I'll make it a little bit faster.
Super excited to be here. Thank you so much for having me. I'm here to share the resources Kaplan has in store for you guys. So let's get started. Let me start by introducing myself. My name is Chada. I'm Kaplan's student brand ambassador here at Baylor. I'm a senior bio pre-med and with a business admin minor here at Baylor. So if you have any questions about Kaplan, MCAT, or anything in general, feel free to reach out to me. I'll be really happy to help. I'm basically linking to you guys, Kaplan, and the organizations of the different clubs like Burst. So yeah, you can always reach out to me anytime, email me, feel free if you have any questions. So let's get started with like some of the discounts that Kaplan offers based off Burst and Kaplan's partnership. So you get a 10% off any course that you buy from Kaplan. You can avail that 10% by using Club 10, the code, at captest.com, or you can call 1-800-CAPTEST. Apart from that, we also have a lot of free events that include free practices. You can attend free practice tests, deep dive sessions about the test, or you can experience how a Kaplan course is taught. All of these events, you can find more information and sign up for them at the link below. But if you have any questions, feel free to reach out to me. So the cool thing we're doing this semester is we're doing a custom-based resources. So based off your journey, you can get different resources. You can use either of the QR code, the snap code QR, or the other QR, or you can use my link to go take you to the homepage. So once you do that, it will bring you to this page here. Here, you will put which year you are, freshman, sophomore, junior, senior, and then you can click on what test you're taking, or your pre-med, pre-law, pre-dent, and then after you do that, you put your first and last name, your email, and then we'll give you a free resource that can range differently for different years. The freshman will get a different resource than a junior or senior because they're on a different part of the journey than obviously upperclassmen for the MCAT. So this basically will help everyone study for the MCAT. On top top of that, everyone will be emailed a study guide. So this is study guide is directly emailed to you. This is a study guide that basically condenses all the material covering the MCAT, but also all the classes that you take. So this is a cool resource. So if you're like want to use it for you after, like before your bio exam or final, or before your chem exam final, or before your sociology or psychology final, it's a cool resource to have. So this will be emailed to you. So be sure to use either one of these links or QR codes or the link to sign up and get at least the study guide, if not the resources. But the resources are also really cool too. Really recommend using this. Okay, now I just want to go briefly and explain the medical school admission cycle. So for the MCAT is one of the main components of it. Most people study for the MCAT about 300 hours, 3 to 4 months, focus more on the hours than months. So that most people study and prepare and take the MCAT either first semester or second semester of their junior year. Apart from taking the MCAT, they also prepare for their personal statements and get their recommendation letters from professors during the second semester of their junior years. Primary applications open on June 1st, and it's ideal to submit it as soon as possible because medical school's admission process is on a rolling basis. That means that once they get your primary, they're going to review it. They're going to review it as they're getting it. So that way, you have a higher chance of getting into medical school if you apply early than late because they're looking at your application as soon as they receive it, and it gives you a higher chance of getting in. After primary, secondary start rolling in, and then secondaries, it's really ideal to submit them within 2 weeks after receiving it. And once you submit your secondary, you start receiving interviews. Interviews start in August and go all the way to February, and they're also on a rolling basis. And as soon as you submit your secondary, you may start receiving interview offers. And then acceptance letters occur somewhere between October and November, and they go all the way to March and even into summer. So this is a brief overline of the medical school admission timeline. So a little bit about MCAT. So MCAT has 4 sections. Apart from the CARS, which is a critical analysis, all of the others are science sections, and they're based off the material that you learn in different science courses that you take here at Baylor. They're all 59 questions, and you get 95 minutes to take it. The CARS is 53, and you get 90 minutes to take it. CARS is like more a reading passage and analysis section. All the others are more science-based. This is a 7.5-hour test. Yes, it's a huge thing. So it's very important that you practice before you take your MCAT because it takes a lot of pacing to do well on the MCAT, apart from knowing obviously the content. So why Kaplan? So here I just want to show a brief video. So now I'm going to share a little bit about the course options. So Kaplan has various course options with various price ranges, and I have some of them listed here. So one of them is test bundles. Test bundles have practice packets, Q banks, and many more, and it starts at $9.99. Apart from that, we have live online, so you can take classes from the comfort of your own home, and they start at $9.99, and they go all the way up depending on the resources you buy. The third option is DIY course, so that way you get all this course and you basically do it on your own. And then lastly, it's tutoring, so that's one-on-one sessions. You sit with an expert and they help you prep, and that starts at $96.99. If you have any questions over which course you should take and like what will fit your study habits, here I have a QR code. You can scan that, and it will take you to the questionnaire, and it will help you guide through it. So someone will help you basically find a course that fits for you. So feel free to use this, and I highly recommend it if you're struggling to find which course will work best for you. Okay, so here are all the resources that Kaplan course includes. Kaplan course has 700 hours of total instruction and practice. They have 14 full exams. They have the Q bank and the MCAT panel. They have all the AMC material available for students to practice on, and also 7 book MCAT subject review books that you can use to review content review. So the MCAT panel. The MCAT panel is basically has live and recorded sessions, and it basically covers the material covered in the book through a video, so people who are visual learners can help and learn the material through that way. And the live sessions, you can ask questions to the teachers, and the teachers are people who have taken the MCAT, have done really well on it, so they do know the answers to most of the questions. And then lastly, there's a Q bank. So the Q bank is basically a customized quizzes that you can take based on how well you're doing or struggling in a course. So if you're struggling in one section, for example, bio, it will like help you find your weak spot and help you like go review more questions over that. But if you're like really good at chem, it won't like make you like just do the same thing over. It'll customize based off your needs. So if you're struggling in one area, it'll help you like do well in that and create questions based off that part. Lastly, Kaplan has a high score guarantee or money backing. So take a baseline before you take an MCAT, before you take a Kaplan course, and then once you take your MCAT, if you score lower than your baseline, you get your money back. You get either your money back, or you can take the Kaplan course once again free of any cost. But if you have any questions regarding this, you can visit the website and it has all information. But you can also feel free to reach out to me if you have any questions too. And lastly, this is for you. Okay, so I'm a senior. I'm about to graduate, so Kaplan needs a student brand ambassador here at Baylor. So if you're really interested, feel free to reach out to me. Some really cool positions, some very flexible. You kind of create your own hours, and then you also get free Kaplan courses that you can use if you're taking MCAT or any other pre-health or even DRE or any of the tests. It's a really cool opportunity. You actually don't need any experience with Kaplan prior to this. It's a really cool opportunity. If you have any questions, feel free to reach out to me, and I can get you started on the process. And here is my email. So thank you so much for having me. But if you have any questions, feel free to shoot me an email. If you see me around, you're welcome to ask me questions too. Thank you for having me once again.
All right, nice. So we'll get started with our announcements. All right, so the member of this week's bye week is Jack Gillies. So Jack is the Journal Club member or leader for the Epidemiology Journal Club, and he's done so much for Burst, and he's also helped a lot of our Burst members and inspired them. And so Jack, if you're here today, we'd really like to show our appreciation and our gratitude for you, and you get one of our Burst prizes later on. So if anyone wants to check out his awesome journal clubs, please do so. This is probably his last semester. So moving on. Okay.
Right, so if you weren't able to make it to our first general meeting, the points requirement is exactly the same as last semester, so 16 points, and it's broken down into 4 different categories. And as always, if you're part of a research lab, you're welcome to fill out the form to earn 6 points of research credit. And we're really excited because there are 2 labs that we're going to feature in our weekly emails, one of which we've already featured in our last weekly email. So Dr. Martin and Dr. Sheasley's labs are looking for undergraduate RAs. So if that's something that you're interested in, just keep your eyes tuned to the emails and you're welcome to reach out to them. And AED, which is kind of like a pre-health organization on campus, they're hosting the 48th annual pre-health symposium. So it's really just a networking event with professional schools all over the country. So it's coming up soon. And if you're interested in attending, we offer 1 point or we offer multiple points depending on how long you stay. So you don't want to miss it. And it's from February 15th to the 20th.
Hi guys, here's the upcoming journal clubs. If you still want to get journal clubs in this week, we have 2 tomorrow. We have neuroscience and epidemiology with Jack, the bi-member of the week if you want to attend. And we also have some more interesting journal clubs coming up. And as always, if you missed the how to read research article and you need help, you can check out this link, which will be sent out when the PowerPoint's sent out, and it'll take you to a video of that section.
Just a reminder that we have a requirement for 4 service hours, which is 4 points. The sign up is here as well as the weekly email. We have a variety of events that are online, such as missing maps or tutoring at La Vega, as well as in-person events like the Humane Society. Unfortunately, the mentor-mentee program has closed for signups, but your mentors should be emailing you soon with mentees' individual meeting assignments. But yeah.
So we have post-meeting social right after this. It'll be really fun. Make sure to come to that and look out for Sunday's weekly email for our first research coffee hour this semester. And Feb 24th, we have a Cameron Park hike. So mark the date and hope to see you there.
All right, so as y'all know, you guys need 1 special event point every semester. So this is the special event for this semester. Heart of Texas is going to be on March 27th from 9:30 a.m. to 3 p.m. More information will come out the closer we get to that date, but mark your calendars, guys, and keep that in mind.
All right, and lastly, for academic events, we have a lot going on for you all. The Curriculum Vitae CV workshop with the BU Career Center representative will be on the 17th, and that is a Wednesday. The next ones are the LinkedIn and the MCAT workshops and lab techniques. All the dates are listed in our Burst Weekly emails, as well as our social media, as well as the Instagram pages. And in March, we have the GRE workshop with Kaplan. And so it's with Eza, the person who you just saw in the video. All right, so I'd like to introduce our guest speaker for today's general meeting. Dr. Kane is the associate professor in the chemistry and biochemistry department at Baylor. He got his PhD at Texas Tech University, and as well as got his postdoctoral fellow at UCLA. And Dr. Kane currently runs a research group which specializes in the synthesis of bioactive small molecules designed with specialized delivery strategies, especially in drug development. So without further ado, I will now hand it over to Dr. Kane.
Okay, can you hear me?
Yes.
Am I on? Okay. Let me share my screen. Go back to the start. All right, so I think you can see the whole thing anyway, right? I don't know if you're just seeing the slides, but that's okay. So thanks for inviting me. We always love to talk about our research. I'm going to tell you, I'm going to go through this pretty quickly, but I want to tell you about some cool stuff we're doing. Undergraduates are an important part of my research group. You know, unfortunately, you all may find that there aren't enough research groups for the number of undergraduates we have here on campus. And that's really too bad. You know, we're trying to grow our graduate program, and as that grows, we'll be able to, you know, have more undergraduates in the lab. I currently have 5 or 6 in my group. So let me just go ahead and get started. Thanks for the introduction. I just really briefly, I went to Texas Lutheran, very small school down there in San Antonio. Got a chemistry degree, never took a biology or a biochemistry class, because I had a bad experience in high school. So I was done with that. Texas Tech, I got my PhD. It was all organic chemistry. Again, never had any biochemistry, never had any biology. When I went to my postdoc at UCLA, though, I was making molecules for potential cancer therapy, which was pretty cool. Neutron capture therapy, that's a whole different story. I won't talk about it now. But that introduced me to some biochemistry and really an interest in biology. And we've kind of stuck with that since I've been at Baylor. Been here, this is my 25th year, finishing up my 25th year. So I've been here long enough to probably make my organic class absolutely the least popular organic class on campus. But that's completely unfounded. You should come take my class because my students do well. This is an old picture of our group, but just to give you an idea, at this point we had, I don't know, half a dozen graduate students and about the same of undergraduates. Since then, Jeremy, he was really a leader in my group. He started at Baylor as an undergraduate. I don't know, can you see my cursor? Yep. Yes? Mm-hmm. Okay, so this is Jeremy. He was a Baylor undergraduate, stayed for his PhD, and he now is at a postdoc at Massachusetts General Hospital and Harvard. He has a joint appointment there, so he's doing really well. A couple of the undergraduates have graduated as well. Blair, Mina, Harold have all graduated. Oh, and let me see. So anyway, we've got more people now. I have a graduate student who's now at UT Southwestern. I'm sorry, he's at Baylor, Dallas. I had one at UT Southwestern, but Jordan is at Baylor. I'm mentoring him with people there. And then Lauren, Ryan, Elena, Juliana, and Jack are the undergraduates that are working in my lab, along with Allie. Allie is a special case. She started in my group the summer before her freshman year and has worked in my group ever since. She's interviewing for graduate schools now, but I consider her more a graduate student than anything else. So we just an idea of our publications, and I just wanted to point out, you notice highlighted in red our undergraduate co-authors on some of these papers. So these are synthesis papers. My training is in synthesis. So in all of these, we made new molecules. This last one down here, this isn't a undergraduate in my lab, but this was actually my first PhD student. So some of y'all may know Dr. Youngcliff. He was my first PhD student, got his degree in my lab. And then I wanted to point out that we've moved to where we're doing quite a bit of biology. So a bunch of our papers have quite a bit of biology in them. And again, you can see undergraduate authors on at least half the papers, if not more. And so I'm going to talk about a little bit of this biology. I'm not going to go through any of these titles. So I'm going to tell you a story about what we're doing, where we're trying to use synthetic chemistry to make transplants work better. And that seems like kind of a strange thing. We're not just making drugs, but we're making drugs or taking known drugs and figuring out ways to deliver them to try to make transplants work better. There's a lot of work in biomaterials. I consider live tissue to be a biomaterial, you know, the kind of the most obvious kind of biomaterial. And so we do our chemistry on the actual living tissue. So transplants, this is the one opportunity you have where you can take a part out of someone and do some chemistry on it before you put it back in. Otherwise, you got to deal with the whole body. So this is an interesting opportunity. We're not working on hearts right now, but that's kind of a cool picture. So where do we get started in this? Well, we got started when I was first here at Baylor. I was working with Genzyme on ways to repair torn meniscus. So probably a lot of y'all have had a torn meniscus or know of people who have torn their meniscus. This is that pad that's in between your knee. It serves as what do you call it? It's cushioning and also lubrication. But the thing that you'll notice about the meniscus, this area out here, this is the red zone or pink zone. There's some capillaries that you can kind of see giving it a little bit of color. But in this part of the meniscus, this is a cross section. We kind of cut a meniscus into pieces. This area, you don't see much pink. That's because that's where all your weight goes. That makes it hard to pump blood into there. Because of that, that tissue in here doesn't repair very well. Okay, most of your tissue, if you tear something, it'll form a scar, it'll repair. The meniscus, no, it doesn't have that many functional cells to be able to do that. Here we were looking at the penetration of a compound that we'd put on the surface. So that yellow is a compound. What we were doing is trying to glue this meniscus together. We were using molecules like the one up above. We made these. Now we make typically as simple a compound as we can get away with, because we don't want to spend our entire life making the molecules. We want to actually have them and be able to do something with them. So this is a pretty simple molecule, but if you shine a light on it, it forms radicals that cause the cross-linking of amino acids and therefore proteins in the tissue. And this is a graph. We did this with a professor in the engineering department where we looked at the bond strength. So you can see these are a bunch of different meniscuses. They're all different. This is some of the frustration with biology. You know, where do we get these meniscus? Well, we went to the slaughterhouse here in town. We got cows' knees just as soon as the cow was slaughtered. We dissected the meniscus out of their knee, and then we did the chemistry on it. And as you can see, every cow had kind of a special meniscus. That's one of the frustrating things in biology. You know, in chemistry, we get bottles of reagents that are 99% pure. There's no such thing as a 99% pure meniscus. Every cow has its own life story, and because of that, there's a lot of variability. Anyway, what you can see is our treated meniscus formed bonds that had some sort of adhesive strength, whereas the control ones, most of them didn't do anything. This one, the light dehydrated it a little bit probably and made it stick together. So we were able to get bonds. We had a lot of fun with this. I stopped doing it a long time ago, but it introduced us to the idea of tissue chemistry. And so we started playing with different kinds of tissue and trying to see what kind of chemistry we could do on the surfaces. And, you know, we're inspired by the fact that if you're an organic chemist, you think of a tissue surface as being a place where you have functional groups. Organic chemistry, we talk about functional groups. So we have lysine amino groups, we have carbohydrates, we have thiols, typically as disulfides, but we can reduce those to get thiols. Here's some more carbohydrate chemistry. I'm not going to talk about all this chemistry. Here's a glutamic acid or something like that, where we can maybe make an amide bond. And finally, here's just something that's going down into the membrane. So a lot of different chemistry that we can do on tissue surfaces. We decided to explore what can we do. And so here's a piece of pericardium. This is bovine pericardium. It's the tissue that surrounds the heart. One nice thing about it is it's the closest thing you'll get to uniform tissue that I've found in an animal. We can get big sheets of pericardium, and we can do chemistry on them. So here you can see we've modified this with a molecule that catalyzes the emission of light. It's a chemiluminescent assay. And we did chemistry. Here you can see different concentrations. You can see a little bit here. So we're studying to see if we could do, how efficient was this chemistry. You can see the spots. We were trying to localize the stuff. In this graph, this is an old slide, but the idea is we could do chemistry on the tissue. We're still doing that. This is a paper that I'm editing right now, probably be submitted before the end of the month. There's Ali right there, as well as a couple graduate student authors. So we looked very carefully at could we modify pericardium and a variety of other tissues. We put it on there in a way that it was stable to where it wasn't cleaved. But right now we're very interested in things that we can actually attach to tissue surfaces and cleave off. And so here we have a molecule that we can release, and we can release with a certain kinetics. And you'll see why that's important in a minute. So we were interested in the tissue chemistry, and I started trying to figure out where to go with it. And I was actually thinking about wound healing, and still think there's some applications in wound healing. But I met somebody up in Dallas who is a transplant scientist, and I got really interested in transplantation. And specifically, I got interested in eyelid transplantation. So islets are these clusters of cells, alpha and beta cells primarily, that are found in the pancreas. And those cells make insulin. They're the things that regulate your blood glucose. And there's a bunch of these islets, but they're little, kind of like little organs that are buried in the exocrine tissue of the pancreas. If you take collagenase, which is an enzyme, and inject it in the pancreas, you can release these islets and they stay as kind of cell clusters. And so my collaborator actually does human islet transplantation. So he takes donor pancreases, digests the exocrine tissue and isolates the islets. And then in an outpatient procedure, he can infuse these islets through the portal vein into the liver of a recipient. These islets get stuck in the capillaries. And well, what are they? They're little pieces of meat that are being bathed in glucose, maybe. If they are, then insulin comes out, and they serve as a little mini pancreas inside of your liver. Now in Europe, this is used for type 1 diabetics. It's still in development, but it is clinically approved. Here, what we use it for is acute pancreatitis. So if you have acute pancreatitis, you have terrible pain that can't be remedied by most, even opioids. And so you end up drug addicted, in pain. And so they take your pancreas out. Well, that cures you of the pain, but it makes you a type 1 diabetic because they've taken out your islets. And so what my collaborator does is they'll take your pancreas out, remove the pain, and then they'll inject your islets back into you. And so you've removed the pancreas, but you didn't become a diabetic. Now, the challenge with this procedure is that these cells don't belong in the liver. They don't belong actually in a lab where they're being isolated by treatment with a strong enzyme. They don't belong in a well, not a syringe, but really an IV bag. They're just really beat up. And when they get beat up, they start becoming very inflamed, causing inflammation in the liver. And we lose about 80% of the islets within the first couple of days after transplant. It's amazing that this procedure works with the loss of that, but that just tells you the redundancy of the islets. And so what we're interested is, can we protect these islets to where we can get 50% to survive. If we could have 50% survive, the cure would be much more durable. So we're going to see what we can do about that. So what we decided to do is try to address this acute loss of islets after transplantation. There are other problems. There's long-term rejection. If you get somebody else's islets, if they're your own islets, that isn't so much a problem. But if there's someone else's, it's a transplant. Even though it's just cell clusters in your liver, still somebody else's cell clusters. So you need permanent immunosuppression. Finally, it'd be nice if we could take pig islets and do this, do xenotransplantation. Then we wouldn't have to have donors except for the, the bacon makers. Xenotransplantation, people are really looking into that, but there are some real challenges, including this acute loss and this long-term rejection. So can we use some sort of chemical modification to protect these islets? Well, so this is what we've been exploring for several years. We've been trying to take these islets, do chemistry on them in a way to where they're modified, and then see if they can help the transplant work better. So here's some pictures of some islets. There's mouse islets, human islets, and pig islets. We can work with human tissue because, again, we're working with a human transplant lab. What we did is we did modifications. So here's 3 different types of modifications, 3 different kinds of islets, and then these are controls. And what you can see, this is a blue nuclear stain, just to show you where the islet is. And the red stain is propydium iodide, which shows you where you have dead cells. And what you can see is, well, there's really not a big change between the chemically modified islets and the control islets as to how many have died. And we can quantitate that and show that that's the case. So we're not hurting the islets by chemically modifying them. Chemical modification, if we do it with a fluorescent molecule, we can see its persistence. So here is a week after modification, we can see that the modifications are still persistent. Now, not with this chemistry. This chemistry, it's more intense. This chemistry, it's actually more persistent. That is, the level of modification is more consistent. So we might take advantage of, hey, this one stains a lot, but it goes away fast. This one doesn't stain much, but it lasts. This one stains quite a bit and it goes away with a certain rate. So we might take advantage of that. So what are we going to do now? Well, one of the big ways that we lose islets in that first part is through innate inflammation. Your innate immune system, the first thing to kind of react to this transplant tissue. And one of the big pathways is through the Tlr4 pathway. Now, you may be familiar with Tlr4. So toll-like receptors. These are receptors that bind non-self kind of patterns. They don't recognize specific pathogens, but they recognize things like flagellin. So flagellin, that means you've got a bacteria trying to swim through you. Okay, that's a signal that, well, there's probably something that doesn't belong. These are some lipoproteins. There's also things like TLR7 and 8 that recognize RNA and DNA. Okay, those are telling you probably have a virus, an RNA virus, or because you shouldn't have RNA or DNA just floating around. So you have all these different toll-like receptors. TLR4 was characterized as an LPS, or lipopolysaccharide, or endotoxin. If you've worked in a biology lab, you'll see things are endotoxin free. This is something that comes from bacteria cell walls, and it's kind of ubiquitous. It just is everywhere, okay? And so it's hard to get rid of, but it turns out it's a real strong stimulator of TLR4. There's a variety of things, though, that stimulate TLR4. HMGB1 is one protein that does it. HMGB1 comes out when you have cell damage. And so cell damage stimulates this pathway. Suddenly you have all this pro-inflammation, and the cycle goes, you get more damage, you get more inflammation, more damage, more inflammation, end up losing cells. So we decided to see, could we block this TLR4 pathway? And if so, would it help the cells? So we found this compound in the literature. This is a pretty simple looking molecule, but it's a very potent TLR4 antagonist. If you're curious about the biochemistry, it binds into the TLR4 receptor, not at the receptor site, it binds in the signaling site, but a cysteine in the receptor does a Michael addition to this alpha beta unsaturated ester, and it's actually a covalent inhibitor. So that's a good inhibitor. So we thought, well, could we deliver that to the transplant? Maybe we could put a linker on it and attach it to something. So we put a linker on it, but what we find is when you modify this site, not surprisingly, this compound's no longer a good drug. We actually knew that before we did the experiments, but just to let you know, this is a good drug, this is not. So, well, how can we do a linker on here? What can we do? Well, we decided to use a strategy where we took the islets, we modified them by immobilizing the drug on the surface, but we immobilized it in a way to where it would slowly be released. So the idea is this transplanted tissue would be protected by being slowly released after transplantation. So what we're doing is we're protecting the tissue by a localized drug that's only around the tissue because that's what it's attached to. And it's being delivered slowly and continuously at the site of transplant. So these are the molecules that we then decided to work with. This is kind of a bad slide, but if we look here at the right, this is the molecule that's important. So we use some click chemistry just for convenience sake. That's this big thing here. We use this strained cycloalkyne. 8-member ring is as small as you can get an alkyne in, but it's strained. It wants to be linear. And so it reacts rapidly with azides. Here's the azide that was attached to our drug. We use that to mobilize our drug to the tissue surface. So here's the drug, and here's a linker. And this linker has something special here. It has this sulfone. What this sulfone does is it modulates the acidity of these hydrogens. And it turns out it makes these hydrogens just acidic enough to where it can be deprotonated, undergo a beta elimination, and release the drug. And it does that by a certain kinetics based on how acidic are these and how basic is the environment. Well, we know that the pH of the environment pretty well, so we can measure kind of the kinetics of release that we expect. So of course we had to do some experiments first. So here's an islet. Here we modified the islet with a linker, and that linker has a dye on it instead of a drug. We wanted to just characterize the chemistry. So there's the islet coated with the drug. You can see the coating is just on the edge of the islet. So just on the exterior of the islet. So we've put a bunch of drug, or in this case, dye, on the outside of the islet. And then what we did is we cleaved that dye off. We measured how much was released. That tells us how much drug is attached to each islet. So we could determine how much we're delivering. And then we measured the release of the drug itself. So here's the release of the drug. And what you see is a good slow release. This is over 3 days. We've had about 40% of the drug come off. But what it turns out is about 20% of the drug has fallen off by a side reaction. So that's unfortunate. We didn't want this side reaction to happen, where this drug is actually cleaving there instead of where it is. So this leads to a bunch of chemical work that we're doing in our lab right now. I tend to talk more about the downstream results, but what we spend most of our time doing is making the molecules. So we're making different linkers that don't suffer from this unsatisfying, unrewarding hydrolysis. But we can see it's a pretty steady release of the drug, at least. Each islet releases plenty, and so it sounds like it could work. But turns out it does work. So here's some transplant experiments. We've got diabetic mice. We make them diabetic by treating them with a chemical that kills their islets. So here you can see their blood glucose is high at day 0. Then we transplant either control islets, that's islets that haven't been modified at all, don't have any drug, or drug-treated islets, that's the green ones, where we've just taken the islets and treated them with the free drug, okay, as a control, or islets then finally in the red, where we've modified them with our slow-release drug. What you can see is that the control islets don't cure any of the mice. They all stayed diabetic all the way through the procedure. The drug-treated ones, they get better, but they're not cured. Down here below 200 milligrams per deciliter is considered the normal glycemia for a mouse. They did better because we protected them during that first maybe 12, 24 hours. We didn't protect them out as far as we would like. If we modified them to where the drug was slowly and continuously being released, what we see is after about 20 days, all the mice have been cured. And that was persistent. And I'm not going to show you slides about it, but it's kind of interesting how you do this. These islets are transplanted to the kidney capsule. Mouse has 2 kidneys. So what we do is we take the kidney out that we put the islets in, and they become diabetic again immediately. So that shows that it was the transplant that cured their diabetes and they didn't just get better. We followed this up quite a bit, so we know quite a bit more about this, but I'm going to move on to kind of some new stuff that we're doing. So that's pretty cool. We can protect islets from being destroyed and make transplants work better by attaching a drug that's then slowly released after transplant to protect the tissue itself. So what are we doing now? Well, I'm not going to go into this slide. I'm just going to go on to them. So I'm going to show you just 2 projects that we're working on. I want to keep this as short as I can. So right now we're working on hepatocyte modification. Just got an NIH grant on this, and it's actually focused on undergraduates doing a lot of this work. So we have a good group of undergraduates in our lab who are really involved in this, and during the summer are going to be really making some progress. So here's a hepatocyte. It's a liver cell. There's a real interest in hepatocyte transplantations. You can isolate hepatocytes from livers. You can actually freeze them and store them. And so you could have a freezer full of liver cells in the hospital. Somebody comes in with liver failure. You could give them an infusion of liver cells out of the freezer and supplement their liver and make it maybe sustain them until they can get a full liver transplant. There's other things you can do. This has really been looked at in pediatric liver disease where patients don't have some specific enzyme function. You know, the enzyme deficiency, PKU is one that you see on soda cans quite a bit. That's an enzyme deficiency. You can take these patients and give them supplements of liver cells that have that enzyme function and actually cure them. So here's the hepatocyte, but again, we have a problem. When we transplant these, 80 to 90% of them die within the first couple of days. So just like we did with islets, we want to see if we can protect them. Here to the right is what we were doing with the islets. We were attaching drug to them that was slowly released. In this proposal, we're looking at 2 other methods of protecting them. One is we're just going to camouflage them. We're going to make this hepatocyte not look like a cell. We're going to try to see if we can get it just to completely look like some biomaterial that's sitting there functioning as a hepatocyte. You got to be able to get nutrients in and byproducts out, or drugs in and detoxified drugs out. And so this is a trick, but we're pretty confident we can make some progress there. We're also attaching a CD47 peptide. That's a don't eat me signal to your immune system. And it tells your immune system, hey, leave me alone. And so we're hoping that if we graft those peptides onto the surface, we'll actually be able to protect these hepatocytes. So we're working actively on all 3 of these approaches. So what do? We take hepatocytes. Here's hepatocytes. There's the nuclear stain. Here we've attached a green dye to the outside. So we've shown that we can do the chemistry. That green dye, this isn't a cell stain. This is actually covalently attached to the hepatocytes themselves. Finally, propydium iodide tells us a little bit about the toxicity, but we look at that toxicity in multiple ways. So here's hepatocytes that have been modified in a variety of ways. These are primary hepatocytes. So they come from an animal. They don't culture. You can't keep them alive other than by freezing them. We do human, we do rat, and we do mouse hepatocytes. So over a period of days, primary cells die. They don't proliferate, so they just die. You can't keep them alive forever. But you can see that our modified ones are just the same pretty much. You can't see which is which, but the control hepatocytes are in that same group of lines as all the modified ones. So we've shown that we can do a bunch of different modifications. We look at things like do these hepatocytes still do metabolism? So here's ammonium. Can you metabolize ammonia? We measure the urea that's produced in that metabolism. Or here's lidocaine. Lidocaine is a drug that your hepatocytes can very actively metabolize. And here we show that, again, the hepatocytes have been modified, do metabolism, just like the hepatocytes that haven't been modified. Down here at the bottom, this is dead hepatocytes. Dead hepatocytes aren't viable. Dead hepatocytes don't do ammonia metabolism or lidocaine metabolism. So here's a kind of a cool experiment. We wanted to see, could we put two different things on the hepatocyte? So nuclear stain, there's one of the colors, here's another color. So we covalently modified them with two different types of chemistry, looking at two different functional groups. There's the propidium, again, looking at toxicity. So here's a merged slide to show, well, looks like some of them just stained with the green. We're trying to understand that. It looks like those are clusters, and actually, it looks like those are dead cells. So we're trying to understand, why are these dead cells labeling so efficiently with this chemistry? I'm not going to describe the different chemistries that we're using. This chemistry doesn't seem to be localized on dead cells. We use flow cytometry to look at a lot of this. So here's flow cytometry. We can modify in this method, or if we do both, it ends up in this quadrant, showing us we have both modifications. Okay, another project we're really interested in is modifying the vascular endothelium. That's a place where you don't have to remove the tissue to modify. You can actually do that inside of somebody. So can we modify the vascular endothelium? We started out working in the working ex vivo. So we would take rat parts. Here's a rat thoracic aorta that was dissected out of a rat. We infuse it with chemicals to see if we can modify it. Here you can see 5 minutes, we get a little bit of modification. Looks like 10 minutes, we get the most modification. These are just as the edges of a thoracic aorta. We've got the whole picture as well. And so we did some in vitro work with that to figure that out. Then we started working with whole parts. So here's a rat hind limb, and we've infused the vasculature, and you can see, and if I could blow this up some, which I can't, you can see there's capillary beds that are here, especially in the ankle, capillary beds where we've modified the vasculature. Here's a cross section. We make a cut and we can look at the vasculature, and you can see that we've modified just the edges of the veins and the arteries. And so we saw that we could do that. What are we going to do with that? Well, here's a leg transplant. So this leg transplant was done with our chemistry down in San Antonio at Fort Sam at the Air Force Institute. So it's a white rat that got a black rat's leg attached to it. We've done now 16 transplants. Each 4 different groups. I had to black out this because I saw you were filming this presentation. I can't show this molecule if you're filming it. But what we're doing is we're modifying the vasculature before they do the transplantation in ways that we hope are going to protect that leg from immune attack. So here's some of the flow cytometry showing the immune activation. We get some single cells, and then we look at some of the markers to see what kind of immune cells have been stimulated. Well, here's another place where we can, actually this would be an excorporeal as well. And the leg infusion is extracorporeal. They take the leg off, we do the infusion, and then we attach it back. We are interested in doing that in vivo as well. Here's a kidney, and so you can see the kidney we've been able to modify, especially in the glomeruli. And so kidney transplant is a big, big problem, and we'd like to try to contribute to that as well. So that's it. I kind of zoomed through a lot of biology or the biological applications, but just a reminder, what we spend a lot of our time doing is we do the chemistry to make the molecules. We needed some pretty complex molecules to be able to have a hope of making this leg survive for a longer period of time. So I think with that, I'm going to stop. Hopefully I didn't go too long, 30 minutes. I kept it pretty short. I can answer some questions if you got them, or I will just turn over the screen to whoever wants it.
All right, thank you so much, Dr. Kane. If anyone has any questions, you can put it in the Zoom chat, or you can unmute yourself and talk right away. And by the way, to everyone here today, especially the freshmen or the sophomores, if you're planning on taking organic chemistry, do consider registering for Dr. Kane's section. Personally, for me, I had a lot of fun in this course, and I really liked Allie's SI sessions. And so I encourage all of you to consider it, and you might also make really long-lasting friendships too. And thank you, Dr. Kane, again.
Sure.
I will also share my screen. So
I gotta stop.
okay, perfect.
And if you don't have questions now, you can always write me an email or come talk to me sometime. The one thing I'd say about undergraduate research is try to get started like now. You know, don't wait until you're a junior or senior. It's really too late. I have trouble with having people who don't get started early in my lab because the learning curve is very steep. So the earlier you can get started with research, the better. Like I said, Allie started before she was at Baylor. So you don't really have to know much to be able to cook. And that's what we start doing.
Awesome. So if anyone has any questions, you can feel free to email Dr. Kane or put it in the chat box. Other than that, I will wrap up with this slide. So if you want to get any real-time updates with burst meetings or any of the workshops, feel free to follow any of these accounts. And I'll head over to this slide. All right, so I will drop the Google form for signing out into the Zoom chat as well. And make sure to stay for the post-meeting social to also get your one event point. And thank you everyone for coming. If you have any questions, also feel free to put that in there in the chat box. Oh, I'm sorry. Okay, so if you're able to use the QR code, go ahead and do that. Otherwise, let's see. Thanks, Angelo. Okay, once everyone's wrapped up, Akshat, I'll hand it over to you. Okay, I think everyone's good to go. I'll stop sharing. Oh wait, someone wrote in the chat box.